And now we have with us Dr. Danilo Carvahlo. And he is from the International Atomic Energy Agency in Vienna. And he has a presentation on lessons learned from sustained field release of transgenic sterile male mosquitoes in Brazil. So, Mr. Carvahlo, if you could go ahead now, that would be just terrific.
Okay, can you guys listen?
Yes.
All right. Yeah, so this work I'm going to present, I'm in the agency now, but there's no relation to the agency. I'm just based now on the international agency. So this work was in collaboration with the University of São Paulo, the biofacility, Moscamed, and Oxitec that developed the transgenic line that we use. So as alternative, we have this genetic control to start reducing the population of the mosquitoes. And basically, they developed this riddle technology that was also used previously at the Cayman Island with a reduction of 80% in the overtrap index. So this technology is based on the SIT, that is the sterile insect technique, using radiation basically. But instead of using radiation, we still releasing the males, and they are not actually sterile. They pass a lethal gene to the offspring. So this is not, for sure, is not considered a gene-driven mechanism. So because we're killing the offspring. Okay. So once that we make it a transgenic, we also insert a genetic marker. So during the monitoring process, it's quite easy to identify when you collect these eggs, hatch them, and put in a fluorescent, under fluorescent light, and you can see them like this, while the wild type will look like these larvae here. So this is the gene that we used. It's quite simple. I'll show you in the next slide better how it works. But basically, this line is tetracycline dependent. What that means is without tetracycline, the gene is active and will produce the TTAAV protein that, through positive feedback, will kill the mosquito or will kill the larvae before reach pupae. And for the colony to keep this line alive, producing eggs in the facility, for example, we feed them with the tetracycline. So this protein is blocked because it has more chemical affinity to this antibiotic than to the rest of the construction. So it avoids the mosquito to die. So the idea is basically also what Dr. Niels mentioned, is release these insects into the environment. They will mate with the females, but instead of giving or establishing a new population to substitute the population, you're going to kill the offspring. So when we do the releases, we release much more wild transgenic mosquitoes compared to the wild type males. This will increase the chances of one of these transgenic males mate to a wild female, and the offspring will be reduced over time. So as I mentioned, this study started in the Cayman Islands in 2010. The Oxitec and the collaborators reached 80% in the overtrap index, and the next phase was to test this technology in a different area. So many aspects considered to use this city, Juazeiro, in the northeast of Brazil. Once because of the presence of this facility, Moscamed, that was already producing sterile fruit flies for agriculture. and also because we have the presence of the mosquito in this area too. But this is not only regarding this technical part, how the line works or how we're going to release the mosquitoes. It also, as Dr. O'Neill mentioned too, the community is important to get involved on that, and also the regulatory part. So before the releases, we also get a contact with the local authorities and the community leaders to see their opinion about this project. And with their help, we could select the areas that we are intending to do the releases. This project with the pre-selected areas went to the CTNBio, that is the Technical National Committee that regulates transgenic in the country. So it involved many ministers and many agencies to discuss about this project. So they put this in a journal, public journal, and it stay for 30 days for people or any kind of organization to get in touch with the CTNBio and ask for more information about this project, or to say, oh, I'm against it, or okay, needs to be discussed more. Once this is done, then the project will be voted with the CTNBio members. Only after that we are able to release the mosquitoes in the field. So this community engagement that we've done previously to the releases is also keep going during these releases and also after these releases, because it's important that the community to understand how the result is going, how the project is evolving. So these 4 areas were the pre-selected ones that we decided to work, but we were only able to do the releases in these 2 areas here. These are the areas. They are around 55 hectares and 33 hectares each. And this is how the neighborhood, these villages look like. When we see the streets, you cannot find easily the breeding sites for the mosquitoes. But when we get inside the houses, this is the picture that we have, a lot of possible breeding sites for the mosquitoes. So we considered in our community engagement that the population had low or no information about the mosquito biology and its behavior, or what would be a transgenic mosquito. But we do know that people could comprehend about sex, about mating, the concept of sterility, and they do know about the mosquito and about dengue. So we use this information, and in a very simple way, we are transmitting these to the concept of the project, saying what we are going to do, what will be our actions. So we split our actions in 3 types. Those that we found that were mandatory, like the home visiting program that we start, local meetings with the community and the politicians also. Also lectures in the schools. The press coverage was very important on radio, on internet, or even the television. Some that we found as recommended actions in local, regional, and national level. We also had an international event during this project that was very interesting to discuss with the international science community. And those that were that we found that could be suggested, like using the social media jingles in days that we are going to release the mosquitoes, so people could be aware when these activity would take place. Also, we spend some time with local events that, or like carnival parades or this kind of stuff to have a stand talking to the people, and of course the international press. So these are some of the activities that we made with the radio, with the community itself, in schools, in the television. So we have this car with loudspeakers, with the jingle, with some spots that with the important message that we wanted the people to fix in their mind. We also distribute some leaflets with very basic information, but important information of how the project works, why we are doing the releases, that we are releasing only males, not the females, and also other curiosities that they could find. The process in the facility is very long, it takes a lot of time, but this chart is to have an idea how it is. So we still have the colony producing eggs, then we have the storage of them, some come back for the colony to maintain them, and some of these eggs go to the batches that are going to produce males to release. So we have these in parallel, the releases that go also for the second phase now that is running for the Jacobina, and the quality control that is in an independent section is still inside the same facility, but different persons are evaluating or checking the quality control of both the colony and the release batches. So this is briefly how it works. Our facility now has a capacity to produce 4 to 5 million eggs per week. As I told, part of these eggs goes to male production, so they keep like these for around 7 days. We get rid of the females in this transgenic system. We cannot get rid before it, so we need to do the sex sorting. So this is what takes more time in our production. Then we take these males, put in these release devices that are just simple pots with 1,000 males each, and then we do release of these males 3 times a week, depending on the area. The number can be changed. So this is the way also we are doing the releases. It's not the best way. We are also working on that to do also pupae releases or aerial adult releases using drones or this kind of technology, even small airplanes. And after these releases, we set up 2 types of traps, one to collect the adults and another one to collect the eggs, the ovitrap. And from these eggs, we hatch them and check for the fluorescence rate in the total. So we can see very easy a wild type larvae from those that are transgenic. And when we start the releases and we reach 50% of transgenic larvae, that's the point when we start the suppression phase. So we keep releasing these mosquitoes into the field, and over time we start seeing the decrease of the population. So in these 2 areas, the studies that we did were a little bit different in each one. We had a mean release rate of 28,000 males per hectare per week during 179 weeks in this red area over here. All of the neighborhood was covered with ovitraps, so we are keep tracking of how the population was developing in these areas. The same was for the second one, for Mandacaru. all the area in this case was treated. We didn't split into a control and a release area. But in this area, we decided to check which could be the maintenance release to see after suppression if I keep releasing mosquitoes, but in a small amount. If I reduce the amount of mosquitoes that I was releasing, if I should able to keep the suppression, if I can keep the suppression in this area. So in this area, we release during 150 weeks. So this is for the monitoring that we are using for both areas. So basically the DG sentinels that we are using, we are counting these adults, splitting in males, females, of course if they were identified, and also checking or counting how many other mosquitoes we found into these traps. All of them were, we use GPS to see and try to find what were the hotspots in these areas and increase the releases close to these areas, probably because they have more breeding size in these points. And the most important one, the overtraps that we got, that we were counting the eggs that we found in the paddles inside the overtraps and measuring the number of eggs per trap. That is one of the measures that we used. These eggs were hatched, and then we established the percentage of fluorescent larvae to check when the point that we are starting to see the suppression phase. And with this collection, we could also establish the overtrap index, that is the percentage of positive traps in result of the total amount of traps that you have. So, as I told you, we were releasing only for this neighborhood, in Taberaba, we were releasing in this red area over here. By the time we also identified that this area or the amount of mosquitoes in the entire neighborhood was too much for our capacity to release. And because we still have a lot of mosquitoes in the control area, they were migrating to these ones. So we decided to reduce, to have a better, to check if the use of this transgenic line could be efficient to show suppression in an open field. So this is the result that we got from this period that we were releasing. We have this historical period over here that is around 6 months, and then we started the releases to see the population going down up to this phase. So we had into the mean historical data from this period was around 38.2 eggs per trap. And during this phase, it was a 70% reduction for 11.4 eggs per trap. And when we stopped the releases and keep monitoring them, we found an increase of the population 22% to 29.9 eggs per trap. In Mandacaru, it was a little bit similar, but of course, it's a tiny area and more isolated, and as we were treating the whole area, was the migration of mosquitoes from another area was more difficult. But even though in the historical period we have 1.97 in the overtrap index, that was reduced up to this period to 0.8, is a 69% reduction. However, when we decided to see the maintenance rate and we decreased the release rate to 8%, so we were around 10,000 males per hectare here, and here is around 2.5, we see an increase of 10%. That means that our decision to, or the percentage of the reduction to this release rate was too low, or it was too soon to do that. On that time, we decided to do because we had a very good period of here of a couple of months that we had almost no eggs being collected in this area. So to summarize, we have a reduction in the overtrap index for Itaberaba of 6% and 70% in the number of eggs per trap. This is for Mandacaru. I'm sorry to change the name here, but it was quite similar. It's 63% in the overtrap index and 59% in the number of eggs per trap. So we could demonstrate that using this transgenic line, we could reach population suppression, but we still need to check maintenance rate to keep the suppression in these areas. So as I said, we still don't know if it was too soon that we decided to decrease, or if 80% was too much. Another study that we did at the beginning, when we were releasing in the entire red area over here, and we were checking all the overtraps, it was to see if we were able to find transgenic mosquitoes outside the release area. And this could be, the reason of that could be two possibilities. One, mated wild females could be flying out of the area, or the released males were also flying out of this area. We are doing the releases on the street, so this red line over here, this is still was the last street that we were releasing. And a previous study that we did of male dispersion, we saw that from the release point that we are doing, the male mosquitoes could fly up to 108 meters from this point. And what we found from the traps that we are checking for fluorescent larvae is that up to 200 meters from the limit of the release, we could find positive overtraps with transgenic larvae. So probably males could be flying out, or females could be flying out of the release area. So our future work that we are investing now is in this new building that we have 450 square meters for the new production to reach to this amount of males per week. This is how the production is going now. We still with some cages that we need to improve them. We are working on that. And the most laborious part of the production, that is the sorting, that is still using the system. My work now in the agency is to see if we can get a genetically sexy strain line, and then we can avoid all this labor of sorting males from females. And this future work that we have now is to treat, because this city called Jacobina, that is close, around 250 kilometers from the facility. Because in the areas that I showed the results previously, it was not possible to see an impact on daily transmission, because there were tiny areas and people could be moving to other neighborhoods in the same city, and we could not see an impact of these releases in the transmission of the virus. So now we decided to, also because of the health secretary of the state of Bahia asked to increase, and he gave us two options. One was the city Jacobina, that is relatively close, and the other one was too far away from around 1,000 kilometers from the facility that would make things quite impossible to be done. So this city was split into these neighborhoods. It's all covered already with overtraps and digit traps to start. It already started actually the monitoring before the releases, and the idea is to start releasing the transgenic mosquitoes, and push it out of the city. So we started with these more isolated villages over here, and now we are still waiting for some budgets or new fundings from the government to increase the production to keep releasing the mosquitoes in the rest of the area. In these two neighborhoods, we already checked the same results that we got in Juazeiro. So I don't have it, but it's also 8% reduction in each of them. So we still have like consistent results between these two areas. So I think that is all. These are people that are involved, mainly who are giving us funds to work with the health secretary of Bahia, and all the work that is done basically with Moscamed is extremely important. They develop all the monitoring system and also the transport that is done now from Juazeiro to Jacobina. So I think that's all.